Usually when we talk about a medical diagnosis, there's this expectation of precision.
Right. Yeah, yeah. Like it's engineering or something.
Exactly. You break your arm, the x-ray shows that jagged white line, and the doctor just points at the screen and says, "There it is! That's the problem."
It's very binary.
Really?
Broken or not broken. I mean, it's clean, and frankly, it's comforting. We like to be able to categorize the damage.
Right. We like to see it. But step into the world of severe physical trauma, and suddenly that x-ray machine is utterly useless for showing you the real danger.
Oh, completely. It is the absolute definition of diagnostic muddy waters.
Yeah.
You can fix the bone, you can stop the bleeding, but the patient is still crashing, and the imaging just won't tell you why.
Which brings us to today's topic. For all of you listening, we're doing a deep dive into an incredibly detailed medical excerpt on systemic response to injury and metabolic support.
It's a fascinating paper.
It really is, because the mystery we're unpacking here is profound. When someone suffers a massive physical trauma, say a severe car crash, you and I intuitively assume the danger is the physical damage itself.
Right. The crushed bone or massive blood loss.
Exactly. Or, we assume it's a secondary bacterial infection setting in days later. But our source material reveals a hidden killer.
And it's one most people never think about.
It turns out the most dangerous threat might actually be your body's own immune system massively overreacting to what is a completely sterile injury.
Meaning no bacteria required at all.
Right. Okay, let's unpack this. How does a completely sterile injury trigger an immune response that can literally be fatal?
So to understand the underlying mechanism, we basically need to look at what actually happens in the trauma bay.
Okay.
There are essentially three distinct trajectories a patient can take after a severe impact. The first group, unfortunately, succumbs almost immediately.
Because of the raw physical damage?
Yeah, overwhelming, catastrophic tissue injury, massive hemorrhage, immediate organ destruction. Though, as pre-hospital care and tourniquet protocols have improved, this group has actually gotten smaller.
Which is great, obviously.
It is. But the real biological mystery, the part that confuses doctors, lies in the second and third groups.
Those are the people who survive the initial impact, get stabilized, but then just deteriorate later, right?
Exactly. The second subgroup survives the initial injury, maybe even looks like they are recovering, but then they die weeks later in the hospital from secondary organ damage.
Weeks later.
Yeah. And here's the kicker. The organs failing are totally remote from the original injury site.
Wait, really? Like what?
Like a crushed leg leading to liver failure or lung failure.
Wow.
And then the third group develops something called PICS, persistent inflammation, immunosuppression, and catabolism syndrome.
PICS. That sounds like the body is just trapped in a perpetual state of warfare.
It really is. They have these extended, incredibly complicated ICU stays. They're constantly inflamed, yet their immune system is too exhausted to fight off real infections.
Oh, man.
And their bodies literally start breaking down their own muscle mass just for energy. They often never return to their pre-injury baseline.
What stands out to me in the source is that these patients who die weeks later or who develop PICS, they don't necessarily have worse physical wounds than the patients who recover perfectly.
No, not at all. Two people can have the exact same crushed femur, but have completely different outcomes.
That's wild. So what's the deciding factor?
The difference between survival and death late in the hospital stay isn't about the severity of the initial bruised lung or broken bone. It comes down to the degree and the duration of their dysregulated inflammatory response.
Okay.
Their immune system essentially initiates a friendly fire event that ravages their own healthy organs.
But this leads to a question that just screams out to me, and I'm sure you're wondering this too as you listen. If there is no bacterial infection, if it is a completely sterile injury from blunt force,
Right.
how does the immune system even know there is a problem? What is triggering this massive whole-body inflammation if there are no invading bugs to fight?
Well, the body have a chemical alarm system hidden inside absolutely every single cell.
Okay.
We call these molecules DAMPs, which stands for damage-associated molecular patterns. They're also, very fittingly, called alarmins.
Alarmins, I love that. It paints a very clear picture.
It does, yeah.
Yeah.
Under normal, healthy conditions, these are just regular, hardworking, endogenous molecules. They live peacefully inside the cell doing their daily microscopic jobs,
Like what kind of jobs?
helping with DNA replication, folding proteins, generating energy, just basic maintenance. But the second a cell is crushed or ruptured by physical trauma, these molecules spill out into the extracellular space.
So they cross a boundary they are never supposed to cross.
Exactly. And once they're outside the cell, they become massive red flags to the immune system.
Because they don't belong there.
Right. The immune system recognizes that these proteins belong strictly on the inside. If they're on the outside, it means the cell has been violently destroyed.
The source material spends a lot of time on one alarmin in particular, high mobility group box 1, or HMGB1.
Yeah, HNGB1 is the best characterized DAMP in this context. Normally, it's a chromosomal protein,
So it's in the nucleus.
deep inside the nucleus. It helps with basic, essential tasks like DNA repair and transcription. It's actually highly conserved across species, meaning from an evolutionary standpoint, it is fundamental to biological life.
And the text says HMGB1 leaks out passively the moment a cell is damaged. And we can actually detect it in a trauma patient's bloodstream within 30 minutes of the impact.
30 minutes.
That's incredibly fast.
The speed of that release is critical because it sets off a massive cascade.
Wow.
Once outside the cell, HMGB1 causes fever, it causes weight loss, it makes epithelial barriers.
Okay, the lining of your gut.
Yeah, the gut or the lungs. It makes them leaky and permeable. It heavily drives what biologists call sickness behavior.
Reading about HMGB1, I couldn't help but think of it as like a cellular Swiss Army knife.
Oh, that's a good way to put it.
Because it isn't just a static alarm bell. Its function changes entirely based on its redox state, meaning how its sulfur bonds are physically arranged.
Yes. The redox state dictates absolutely everything it does once it's outside the cell. It's a highly dynamic molecule.
So if it's a Swiss Army knife, the configuration of the blades completely alters its use. Our source details this beautifully.
It really does.
Like if HMGB1 is fully reduced, meaning no sulfur bonds have formed, it acts like a chemical magnet, right?
Right, it just attracts other immune cells to the area.
But if a specific bond forms between two cysteine amino acids, it flips open a new, highly dangerous blade.
And that's when things go wrong.
Yeah, it gains these incredibly potent pro-inflammatory properties, prompting immune cells to release a storm of inflammatory chemicals.
And then, if the environment becomes too oxalized, it folds up all its blades and becomes completely inactive.
It literally shapeshifts based on the chemical environment around it. That's crazy.
It is. The body is constantly regulating this shapeshifting to try and control the inflammation. In human trauma patients, HMGB1 levels correlate directly with the severity of their injuries and their subsequent organ failure. In animal models, if researchers just inject pure HMGB1 into a healthy subject, that subject develops fever, leaky blood vessels, and can even die.
Just from that one protein being placed in the wrong location.
Just from that.
Okay, so shape-shifting proteins sound the alarm. But what happens when the alarm itself looks exactly like an ancient enemy?
Ah, yes.
Because there is another damp that is honestly even more mind-bending to me. It doesn't just ring a bell, it actively tricks the immune system.
What's fascinating here is that we are talking about our own mitochondria.
The powerhouses of the cell.
Exactly, generating our energy.
Yeah.
But from an evolutionary perspective, they originally evolved from bacteria.
Which is still so weird to think about.
I know, but billions of years ago, free-living bacteria were engulfed by our cellular ancestors, and rather than being destroyed, they formed a symbiotic relationship.
But because they used to be bacteria, they still carry bacterial features.
Right down to their DNA. Mitochondria have their own distinct genetic code mitochondrial DNA, or mtDNA.
Okay.
And to our modern immune system, this DNA looks distinctly bacterial. It lacks certain protective coatings that our regular nuclear DNA has.
This completely blew my mind. Think about the scale of this for a second. If you are in a car crash and a muscle is severely crushed, millions of your cells rupture.
Millions.
And every single one of those cells contains hundreds or thousands of mitochondria.
Right.
They're all destroyed simultaneously, releasing this cell-free mitochondrial DNA directly into your bloodstream.
The volume is staggering.
Oh.
Cell-free mtDNA levels in trauma patients can spike thousands of times higher than normal.
Thousands.
Yeah. And because it retains those ancient bacterial molecular signatures, the immune system doesn't just see tissue damage. It thinks the body is experiencing a massive, overwhelming bacterial invasion.
Even though there isn't a single actual bacterium in sight, the physical trauma just popped the balloons holding our evolutionary past.
It's exactly it. It triggers the innate immune system to launch a devastating, unnecessary attack.
It's fighting a ghost.
A phantom infection. Yeah. This involves activating complex pathways like the Esting pathway. The immune system goes to all-out war. And the collateral damage of that war is remote organ failure.
So that's how a crushed leg hurts the lungs.
Right. The heavy artillery the immune system fires off can severely damage the lungs and the liver, completely far away from the initial site of impact.
I mean, the body's ancient evolutionary history literally turns against it in the modern ICU.
It really does.
So we have DNA and DNA repair proteins causing chaos.
Mm.
But what about the cell's own repair crew? Like, if a cell is being crushed, it's going to try and fix itself. Do those repair proteins also turn against us if they leak out?
They absolutely do. We see this with heat shock proteins, or HSPs.
Okay.
These are molecular chaperones. When a cell experiences stress, like a sudden spike in heat or oxygen deprivation, HSPs act like emergency medics.
Right.
They make sure other proteins don't misfold under pressure. They refold damaged proteins or tag irreparably damaged ones for destruction. They basically keep the cellular machinery running smoothly under duress.
But just like HMGB1, if they leak out of a massively damaged cell, they become inflammatory damps, right?
They do, but there is a really subtle, elegant nuance to how the body handles them. The immune system's reaction depends heavily on how the HSPs arrive outside the cell.
What do you mean by how?
Well, if they leak out passively because the cell was violently destroyed, they bind to receptors that scream danger, causing widespread inflammation.
But they can also be actively secreted by intact cells that are just stressed, right? Not dead, just struggling.
And that changes everything. If a stressed but living cell actively packages these proteins into tiny bubbles, called exosomes, and secretes them,
Okay.
they can actually provide immune-dampening anti-inflammatory signals to the surrounding tissue.
So it's not always an alarm.
Exactly. It's like the immune system has a volume knob. For instance, if an extracellular HSP binds to a receptor called Siglec-14, it turns the volume up, triggering inflammation.
But there's another one, right?
Yeah, if it binds to a nearly identical receptor called Siglec-5, it turns the volume down, triggering an anti-inflammatory response.
It's incredible how context-dependent every single molecule is.
Hmm.
But wait, I need to stop you there because the next trigger the source brings up just defies biological logic to me.
But let me guess, HEMA.
Heme. Heme is the oxygen-binding part of hemoglobin in our red blood cells.
It is the core component of our oxygen transport system.
Hemoglobin is what keeps us alive. It carries oxygen to every cell in our body. How can the very thing giving us breath suddenly become a toxic alarm bell?
It comes back to that concept of being in the wrong place. When red blood cells are crushed and burst in a blunt trauma, raw hemoglobin is released into the blood plasma.
Okay.
Normally, we have scavenger proteins floating around that act like a cleanup crew. They bind up loose hemoglobin and take it to the liver to be safely broken down.
But in massive trauma, there's just too much of it.
Way too much. The cleanup crew is totally overwhelmed.
So what happens to the leftover hemoglobin?
It oxidizes, and it drops its oxygen-carrying component. It releases what we call label heme, and label heme is a highly reactive pro-oxidant.
Oh.
Yeah. Unchaperoned in the bloodstream, it acts as a potent damp.
What kind of damage are we talking about here?
It is directly cytotoxic. Label heme physically interacts with the membranes of healthy cells and catalyzes something called lipid peroxidation.
Meaning what, exactly?
Imagine a protective layer of fat surrounding a cell, and the heme acts like an acid, chemically degrading that fatty layer,
It melts it.
basically. This physical destruction triggers a highly inflammatory form of programmed cell death called necroptosis.
It literally melts the armor of surrounding cells. It goes from life giver to cell destroyer just by slipping out of its designated lane.
It's a stark reminder that in biology, safety is entirely about compartmentalization.
Speaking of compartmentalization, we have to talk about the physical structure holding all these cells together. The extracellular matrix, or the ECM.
The scaffolding?
Yeah, the ECM is a dense web of proteins and sugars that gives our tissues physical structure.
Right.
And bound up within that scaffolding is a molecule called biglycan. When tissue is injured, enzymes rush in and chop up the damaged scaffolding, which releases biglycan into a soluble, free-floating form.
And this is where the source highlights a major, terrifying anomaly.
It really is an anomaly. Usually for an immune cell to release a very specific, highly potent inflammatory chemical called IL-1, it requires two completely separate biochemical signals. It's a strict fail-safe mechanism.
Think of it like a nuclear missile silo. You need two separate keys turned simultaneously to launch the weapon.
Right.
One signal tells the cell to manufacture the raw material, and a second separate signal activates the machinery to process it into its dangerous explosive form.
But when that cellular scaffolding breaks, Soluble biglycan bypasses the failsafe.
Entirely.
It uniquely provides both signals all by itself. It hits one receptor to start the manufacturing and simultaneously hits a different receptor to activate the weapon.
It single-handedly unleashes a robust inflammatory response.
Right.
It's a highly efficient, localized alarm.
The collapsing building itself becomes the siren.
Exactly.
So we have shape-shifting proteins, evolutionary ghosts from ancient bacteria, toxic oxygen carriers, and broken scaffolding all screaming at the top of their lungs that the body is under attack.
It's a lot of noise.
But who is actually listening to them? How do the immune cells hear these alarms floating in the blood?
They hear them through a family of cellular sensors called pattern recognition receptors, or PRRs.
Okay.
These are the literal ears of the innate immune system. And the most famous of these are the toll-like receptors.
I love the trivia about where that name comes from. These were first discovered in fruit flies, right?
Yeah. A researcher looking at fruit fly mutations found a gene that, when altered, made the flies look incredibly bizarre, prompting the German researcher to exclaim, "Das ist ja toll."
Which translates to, that's crazy or that's amazing.
Exactly. Later, scientists realized these toll receptors were crucial for immune defense against fungal infections. Humans have a whole suite of these toll-like receptors.
And they sit right on the outer surface of our immune cells waiting to detect these dams.
Waiting for the alarms, yeah.
But how does a receptor on the outside of a cell tell the nucleus deep on the inside to start pumping out inflammatory chemicals?
It's like a microscopic bucket brigade. You don't need to memorize the dizzying list of protein names. What matters is the physical chain reaction.
Okay, walk us through it.
When an alarm like HMGB1 hits a receptor on the outside of the cell, the receptor physically twists and pairs up with a neighbor.
Changing shape again.
Right. That physical shape change recruits an adapter protein inside the cell. That protein activates a kinase, an enzyme that acts like a catalyst, which clips another protein, which activates another.
It's a cascade of dominoes falling.
Exactly that. And by the time this chain reaction reaches the nucleus, it destroys a cellular lock, freeing up a master switch.
And that switch
Enters the nucleus and turns on the genes that scream, "We're under attack," manufacture inflammation.
And that's just the receptors on the outside surface. The source goes into incredible detail about the intracellular receptors, the alarms waiting inside the immune cells.
Yes, specifically something called the inflammasome.
The inflammasome.
The inflammasome is an absolute marvel of cellular engineering. It is a massive multi-protein complex that lives deactivated in the cytoplasm.
Deactivated?
Yeah, it's basically folded in on itself in a state of self-repression. Imagine a bear trap that is set, but with a safety pin holding it open,
Just waiting for a trigger.
waiting for a damp that has been internalized by the cell.
Yeah.
When the sensor detects that danger, it pulls the safety pin.
Oh, wow.
It unfolds. It then binds with other adapter proteins, and they link together into a massive wheel-like structure,
And when that wheel locks together, it auto-activates an effector protein called caspase-1.
which acts as molecular scissors. Caspase-1 takes those raw, inactive inflammatory chemicals we talked about earlier and rapidly snips them into their mature, highly dangerous forms, ready to be secreted.
It's terrifying but amazing.
The inflammasome is a beautifully precise, yet highly destructive piece of machinery.
It is literal microscopic warfare. But if all of these alarms and bucket brigades and inflammasome wheels are happening on a localized microscopic level, how do doctors actually use this invisible war to help the patient lying in the hospital bed? Like, how does this translate to survival?
That's where we look at how these microscopic battles spill over into the whole body, serving as measurable biomarkers in the blood.
Okay.
By tracking these systemic messengers, doctors can actually look into the future of the patient's recovery.
Let's talk about the S100 proteins here, because this is where it gets highly practical for diagnostics.
S100 proteins are calcium-binding proteins. Like the others, they are released from damaged cells. One specific type, S100B, is highly concentrated in astrocytes, which are structural cells in the brain.
And this is huge for predicting traumatic brain injuries.
Enormous. If a patient comes in with a severe head injury, S100B is a hypersensitive marker that can be measured in their blood within just 3 hours of the injury.
3 hours.
Yeah. The levels of S100B on day 1 significantly predict whether that patient will have a favorable neurological recovery 3 months later, or if they will succumb to the brain injury.
It's essentially a molecular crystal ball.
It really is.
We also see this predictive power with a group of proteins called pentraxins, right?
Yes, pentraxins. These are soluble molecules that float in the blood and act like a targeting system, binding to cell debris. The classic one is C-reactive protein, or CRP.
Anyone who has had blood work for inflammation has probably seen a CRP level.
Right, it's a very common test. When there is systemic inflammation, the liver gets the signal and pumps out CRP, increasing its levels in the blood up to 1,000-fold.
Wow.
It's a massive generalized alarm bell from the liver. But there's another contraction called PTX3.
And PTX3 is unique because it isn't made in the liver at all.
Exactly. PTX3 is synthesized directly at the site of injury by the local peripheral tissues and immune cells. It gives you a real-time read on the local battlefield.
Okay, so what does it tell doctors?
In polytrauma patients, PTX3 peaks at 24 hours. And the data shows that the higher the concentration of PTX3 at that 24-hour mark, the lower the patient's probability of surviving the coming weeks.
So what does this all mean for you, the listener? Why does mapping out these shape-shifting damps and toll-like receptors and pentraxins actually matter?
It matters because of how it changes treatment.
Right, because this isn't just biological trivia. For decades, trauma medicine was purely reactive. Stop the bleeding, fix the broken bone, give antibiotics, and then just wait to see if the patient crashes.
Exactly.
But by understanding this invisible, sterile, inflammatory response, doctors can shift from reacting to the crash to predicting it before it happens.
It's moving from anatomical medicine to proactive molecular medicine.
Yeah.
If we can measure HMGB1 or S100B or PTX3 early on, we can identify the patients whose immune systems are secretly gearing up for a friendly fire assault.
Before the remote organs even start to fail.
Exactly. And if we can block the specific receptor that mitochondrial DNA is binding to, we might stop the remote organ failure before it ever begins. It's about preempting the body's own worst instincts.
It is an incredible journey this source material takes us on, from the sheer physical violence of the initial trauma down to the microscopic release of shape-shifting proteins.
It really connects the macro to the micro.
It does. We've seen how our own crushed mitochondria trick our bodies into fighting a phantom bacterial invasion. We've explored how cellular bucket brigades and inflammasome wheels act as the triggers, escalating local tissue damage into a systemic whole-body war that dictates whether a patient lives or dies weeks later.
It completely redefines what it means to be injured.
It really does. And I want to leave you with one final thought to mull over. We've learned today that much of our body's response to trauma, like mistaking our own mitochondria for ancient bacteria, is deeply hardwired into our evolutionary past.
Millions of years of programming.
Right. These violent immune mechanisms evolved over millions of years to help us survive minor scrapes, animal bites, and local infections. The kinds of things our ancient ancestors face.
Things that could actually survive without an ICU.
Exactly. But today, modern medicine can keep a human body alive through catastrophic, massive trauma that no early human could have ever survived. The rules have completely changed.
The body doesn't know what to do with that level of damage.
It doesn't, which raises a fascinating medical question. As we push the boundaries of human survival, will the next great frontier of trauma medicine require us to intentionally hack and temporarily disable our own evolutionary survival instincts just to keep us alive?
It's a profound thought. To survive the modern world, we may literally have to save us from ourselves.
We may indeed. Thank you so much for joining us on this deep dive. Keep asking questions, keep looking past the obvious, and keep wondering about the incredible microscopic machinery operating inside you every single second.